In this video, I am going to discuss about basic tools of recombinant DNA technology. First is isolation of insert DNA or gene of interest. So this is our host cell that contains the gene of desired function, such as islets of Langerhans of pancreas that secretes insulin. So this cell is disrupted mechanically by the enzyme protease, lipase, amylase and ribonuclease. After that we get our DNA. अब इस डीएनए में रेस्ट्रिक्शन एंजाइम ऐड करने पर यह रेस्ट्रिक्शन एंजाइम उस डीएनए के स्पेसिफिक सीक्वेंस पे जाकर कट लगाता है और ये डीएनए के अलग अलग जगह पर ये सीक्वेंस होता है तो हमें वेरिएशन मिलता है डीएनए फ्रैगमेंट्स में और इन्हें रेस्ट्रिक्शन फ्रैगमेंट भी बोला जाता है विच वेरीज स्पीसीज बाय स्पीसीज एंड दिस वेरिएशन in length of restriction fragment is called restriction fragment length polymorphism. अब जिस फ्रैगमेंट में हमारा जीन है उसे जेल इलेक्ट्रोफोरेसिस टेक्निक के जरिए उससे हम जीन को अलग कर देते हैं. So the second is restriction enzyme. These are also called the molecular scissor and these act as foundation of recombinant DNA technology. These enzymes are present in bacteria and provide a type of defense mechanism called restriction modification system. There are two components present in this restriction modification system. First is restriction enzyme and second is modification system. So here this is our restriction endonuclease. What it does, it recognizes a specific DNA sequence and cut any strand which possesses that sequence. The term restriction refers to its function because it restricts the foreign DNA of bacteriophage and degrades them. सो ये जो स्पेसिफिक सीक्वेंस है वो बैक्टीरिया के डीएनए में भी हो सकता है. सो ये एंजाइम इस बैक्टीरियल डीएनए को भी कट कर सकता है बट ये नहीं हो पाता बिकॉज ऑफ दिस एंजाइम मिथाइल ट्रांसफेरेज. व्हाट इट डज, इट मॉडिफाइज द बैक्टीरियल डीएनए बाय एडिंग methyl group to one or two nucleotides. Therefore this restriction enzyme fail to recognize and cannot degrade and fails to degrade the modified bacterial DNA. So there are three types of restriction enzyme. First is type 1, type 2 and type 3. Type 1 this enzyme cuts DNA at random sites and it requires magnesium ion and S-adenosyl and it use ATP as cofactor. Second is type 2. It is simple and require no ATP and it cuts DNA within recognition site. What is recognition site? It is a sequence of nucleotide which are recognized by restriction enzyme and it consists of 4 to 8 base pair. Here is our type 3. This restriction enzyme cut the DNA about 25 base pair from the recognition sequence and it requires ATP as source of energy. So here type 1 and type 3 requires ATP but type 2 does not require ATP, so it is highly used as recombinant DNA technology.